To perform a dilution of copper amine, begin by carefully transferring a small amount of the stock solution into a clean container for pipetting. Using your pipette, draw 3 milliliters of the stock solution. Check the meniscus, the curved surface of the liquid, and make sure the bottom of the curve aligns as closely as possible with the 3 milliliter mark. Next, transfer the solution from the pipette into a graduated cylinder to confirm the volume still reads 3 milliliters. Once verified, pour the solution into a 25 milliliter volumetric flask. Using a transfer pipette, slowly add distilled water to the flask until the bottom of the meniscus reaches the 25 milliliter calibration line. Be careful not to overfill. Once the volume is correct, you may now transfer this first dilution into a clean beaker to prepare for the next step. For the second dilution, pipette 3 milliliters of your first diluted solution. When measuring, make sure to read the meniscus properly. The meniscus is the curved surface of a liquid caused by the liquid's attraction to the container walls. Always align your eye level with the container and read the lowest point of the curve for an accurate measurement. After confirming your reading, verify the volume in a graduated cylinder, and transfer the solution into a clean volumetric flask. Slowly add distilled water using your transfer pipette until the bottom of the meniscus rests exactly at the 25 milliliter mark. Also, before beginning any laboratory experiment, make sure you are wearing proper safety attire. Once your volume is verified, transfer this second dilution into another clean beaker. For the third dilution, use your pipette to draw 3 milliliters from the second diluted solution. Check your meniscus, confirm the volume in a graduated cylinder, and transfer it to a clean volumetric flask as done with the previous dilutions. The stock solution is a concentrated form of a chemical solution that is being used for the dilutions. A dilution is made by adding solvent, usually distilled water, to a measured amount of the stock to lower its concentration. The concentration and volumes of these solutions are used in the dilution equation, which works because the moles of the solute remain constant while only the volume changes. This allows for a calculation of a specific dilution concentration. Once the diluted solution is transferred to the flask, you may add distilled water to the calibration line without exceeding it. After confirming the final volume, pour this third dilution into another clean beaker. You have now completed 3 successful dilutions of your copper amine solution. These solutions are ready for the next step in your experiment as instructed by your lab supervisor. Finally, clean all glassware and equipment thoroughly, and make sure your workspace is tidy before leaving the lab. Remember, always leave your station cleaner than you found it.